today we'll be talking. My name is Yusuf Ramadan, and today we'll be talking about a scientific phenomenon that may have very important implications in the future. What? Oh, nothing. It's just that, my bad. I'll let you finish in a second, but today we will be talking about cloning. Now, before we begin, what are clones? Well, you see, clones are two organisms with perfectly matching DNA. You can actually see clones today in our natural world with identical twins. Identical twins are created in the first few days of an embryo's creation. Oh, hey, catch! After the sperm fertilizes the egg and the embryo is formed, the embryo then splits in half, and each half keeps dividing on its own, creating two genetically identical individuals. Now that we know all this happens naturally, let's talk about how it can be done artificially by scientists in a lab. You see, artificial embryo tuning is the same process we just went over, but done, once again, by scientists artificially. Scientists take the embryo themselves and separate it in a petri dish, and these two new embryos are then put into surrogate mothers to be fully grown. However, there's another type of cloning apart from the reproductive cloning we just discussed, and that is gene cloning. Gene cloning consists of taking a gene from one organism, which is often referred to as foreign DNA, and inserting it into a vector or plasmid, which are circular pieces of DNA that can come from bacteria, yeast, or viruses and act as carriers of this said foreign DNA. After the gene is inserted, the vector is then placed in laboratory conditions that allow it to multiply. Researchers often do this to introduce genes they want to study into model organisms such as, let's see, lab mice. Now let's talk about the other way cloning can be done artificially by scientists in a lab. Safety goggles on. I think I got it this time. Somatic cell nuclear transfer. Let's break it down. A somatic cell is, first and foremost, any cell that is not reproductive. Nuclear refers to the nucleus where a cell's DNA is held. This DNA is condensed into little packages called chromosomes, which contain the info that makes us, well, us.
Now, on to transfer. One of the most famous cases in which somatic cell nuclear transfer was used was back in 1996 with Dolly the sheep. You see, scientists isolated an egg cell and a somatic cell from a female sheep, and then they removed all the contents of the egg cell's nucleus. They then replaced it with the DNA from the somatic cell's nucleus, and this new fertilized egg, after a few chemical tweaks, developed into an embryo, and this was implanted into a surrogate mother and later carried to term.
Some uses of cloning include increasing agricultural yields, finding out about rare genes that cause diseases, and even organ replacements. And that is the science of cloning.